I was at this meeting a couple of years ago, and every year I try to add a little bit more from, our research add-on. And I apologize if any of you heard me 2 years ago in Denver. There will be some repeat of what I described before, but as Larry suggested, he told me to give the big picture. And that's what I like to do too, because as a parent myself, I want to hear the big picture, not just a tiny piece of the story. How many of you, first of all, are parents? Okay, great. And how many of you are professionals, care providers? That's wonderful. And I think that there's an overlap, too, between those of us who are parents and those of us who are somehow working in the field or working with people with autism spectrum disorder. And that's what brought me to this field of research about almost 10 years ago now. I was at a state where my son was diagnosed almost 25 years ago in the dark ages with respect to autism. And it was something so puzzling and so devastating, that diagnosis. PDD-NOS, which was not even in the medical literature because I looked, couldn't find it. So I started reading about autism, which is really depressing. Imagine 25 years ago. And you're thinking, oh my God, it's a lifetime of just not being able to do very much. But, I think the picture has changed radically since then, and there's a lot of reason to be hopeful for the futures of our children now. So, so what I want to do today is talk about what most of you have in mind, the question, which is what causes autism? And as you would expect, there's not a simple answer to this. But, there's reason to try to understand the underpinnings, the biological underpinnings, because how else can you adequately treat it? The situation with me and my son was that from the age of 6.5 to about 21, 22, he was prescribed almost 20 different medications, none of which produce any positive effects. A lot of negative side effects, as probably many of you parents have seen with your children, but nothing that I could say was a wow effect, like, boy, this medication really, really helps. And, these are all, starting with Ritalin, and working your way up to the atypical antipsychotics, these are all very powerful drugs that have the potential to do a lot of damage, especially during the years of development. And we don't know what happens, when we give these drugs, especially to children. So there's good reason to try to understand the underlying biology of autism so that we can more adequately treat it. Okay, so to this group, I don't really have to go much into detail about what is autism. It's a neurodevelopmental disorder. The DSM-5 has now focused on two of the major symptoms, core symptoms, which are reciprocal social interactions, which in my opinion, is the most handicapping of the core symptoms, because as humans, we need to interact with other individuals. And the fact that social interactions are impaired really handicaps people with autism spectrum disorders, including my son, who's at age 27. He still lacks that ability to naturally integrate with his peers. The other core symptom that is a focus of this diagnosis are the restricted interest and repetitive behavior, stereotype behaviors. And then, of course, along with some of this, with these two, there's often impairments in language development and usage, particularly pragmatics, the use of language for social communication. But the important thing here is that autism is not a single disorder. It's really a spectrum of disorders, so we can't lump it all together in studies to try to understand what is the underlying cause, because it's likely to be multiple different causes. So aside from causes, we know that autism is a significant public health problem. The recent CDC estimate of the prevalence of autism is now risen to 1 in 68. A year ago it was 1 in 88. Before that it was 1 in 110. When I started my research on autism about 10 years ago, it was like 3 to 4 in 10,000. That's a huge increase in the prevalence. Aside from that, there's enormous lifetime costs associated with that. 3.2 million per individual as estimated in about 2006. I'm sure it's a lot more now. So let's get to the meat of what I'm going to be talking about, which is the question of what causes autism. So to try to understand, to attack it from the research point of view, what I did was construct what I call this pyramid, where I'm including in the pyramid all of the factors that need to be considered in order to answer that question. And the pyramid is constructed also by layers. It's sort of a hierarchical representation of all of the factors need to be considered that are ordered in terms of the higher levels controlling everything below it. So there is kind of a rationale for the way that this is constructed. At the bottom layer, I put the phenotypes, the ASD phenotype. So what is that? A phenotype is what is observable. And this could be the behaviors, the symptoms, even the brain circuitry, which is observable using MRI, magnetic resonance imaging techniques. And above that, if you ask, what is directly responsible for those phenotypes? Well, the answer is it has to be the gene expression profiles, which describe the gene activity levels in each cell in every tissue. Okay, so that describes the gene expression or gene activity levels determine what the functions of the cells in the tissues are. So obviously, it's the most direct, it has the most direct impact on the phenotypes. If you move up a layer, side by side, I've put genetics and epigenetics. So what is genetics? I think you've heard a lot about genetics in the literature, genetics of autism. And what genetics refers to are specifically the sequence of DNA in our genome. And the DNA is the same in every cell in your body. Okay, a genetic mechanism involves a change in the order of the four bases that make up your DNA. And the changes can involve, for example, a mutation of one base to another, as indicated by change in one color, like from the yellow on the top line to a green. So that would be a mutation. It could involve a deletion where some parts of the DNA are deleted. So that would change the sequence of the DNA, or they could be duplicated. So there are many different genetic mechanisms, but all of them lead to a change in the sequence of the bases in the DNA. Side by side with genetics, I have what's called epigenetics, which is like the software. These are the readers of the DNA sequence. In other words, the DNA is just there. It's like a blueprint. But what reads the blueprint and actually constructs the proteins that are used in your body? Well, it's dependent upon epigenetic mechanisms. So what does that mean? Epigenetics simply means aside from genetics or outside of genetics. And epigenetic mechanisms, importantly, do not involve a change in the DNA sequence as shown there. However, it can involve a chemical mark placed upon the DNA, but this mark is reversible. So epigenetic changes can be reversible, and that's important. There are several different mechanisms of epigenetic regulation of DNA, and among them DNA methylation, which I will be talking about today, but also a few others like microRNA expression and so forth. And you don't have to know the details. Just know that epigenetic changes don't involve changes in the DNA sequence, and they could be reversible. Okay, and above it all, I have the great unknown. And what are these? These are environmental triggers. Okay, and these triggers can be either intrinsic, that means they could be part of us, physiological type triggers, such as inflammation, hormone imbalances would be physiological triggers, or they could be external environmental factors, such as, for example, pesticides, and it's something we're studying now. So what we're interested in my lab are several things. First of all, we would like to identify the genes and biological pathways or functions in order to develop targeted therapies for that. In other words, let's identify the problem and then try to fix it. But if you don't know what the problem is, how do you fix it? Okay? Current medical approaches of throwing random psychotropic drugs at people, I think, really is not very successful. Secondly, we'd like to identify diagnostic biomarkers for autism, because currently all diagnosis is dependent on behavioral analysis, behavioral evaluations. And I think all of you know that the observations of behaviors can be very ambiguous. It's kind of behaviors are seen in the eyes of the beholder, so it depends on how you report the symptoms to the doctor that will influence his diagnosis, his or her diagnosis. Third thing is we'd like to, because autism is such a complex problem, we'd like to develop a systems level, broader understanding of the pathobiology of autism. In other words, we don't think, and as you'll see later, that it's purely a brain disorder, but rather a systems disorder. So what is the problem with all forms of autism research? Well, the problem is the heterogeneity. That means the diversity of the symptoms that are manifested by individuals with autism. And I represent that heterogeneity by a bowl of mixed fruit. Okay, if someone said I'm going to study autism, but they lump all the cases of autism together into one pot called cases and try to study it, well, what are you really studying? Are you really studying an apple, an orange, a banana, et cetera, et cetera? I mean, it's impossible. It's like digging your hand into a fruit bowl and describing what a fruit feels like. It depends on what you pick out, right? Okay, so how do we deal with this problem of heterogeneity? What we try to do, first of all, from the beginning, is to reduce the clinical heterogeneity using scores on a commonly used so-called gold standard behavioral diagnostic test. Now, that's the best we have right now. And so we, at the starting point, when I started this research almost 10 years ago, we realized that we needed to somehow simplify the cases that we're looking at by clustering them into more similar clinical subgroups. Okay, so the underlying hypothesis of our work is that autism is a systems disorder with severe neurological manifestations, and therefore can be studied using more easily accessible tissues, such as blood, which may reflect the molecular and functional abnormalities in the brain and thus also reveal biomarkers for diagnosis. As I mentioned earlier, our strategy is that of divide and conquer. We try to reduce the heterogeneity among the cases using scores on this commonly used diagnostic test, behavioral diagnostic called the ADIR, to subgroup individuals according to the severity of their behavioral or clinical symptoms. So what is this test? I don't know how many of you parents have had your kids have gone through this autism diagnostic interview. Have any of you? Okay, because it was not available when my son was diagnosed. It only came into being in 1994. So anyway, this is so-called the gold standard autism diagnostic, but it's behaviorally based. So it's a neuropsych asking a parent or primary caregiver a series of about 100 different questions about the child's functioning in different areas, such as spoken language. Questions involve asking about the child's ability to chat, whether or not he or she exhibits echolalia, pronominal reversal, etc. The child's ability to engage in nonverbal communication, such as gestures and head nodding, social functioning, interactive play, the ability to offer comfort, repetitive stereotype behaviors such as rocking, flapping, lining things up, the things that are so visibly obvious, restricted interest, perseverance on topics, or playing with objects, etc. And also, interestingly, savant skills, which include superior visual spatial skills, memory, reading, music, art, and computational skills. And the savant skills, I think, are very, very interesting because as a group, people with autism spectrum disorders have about 10 times higher frequencies of savant skills than the general population. So I think this is something that can be utilized productively, positively, to help the individuals. Okay, so how do we use these scores from this behavioral test? As I said, there are about 100-something scores, and sometimes the questions are asked in two forms. So we've taken a total of 123 scored responses from this test in order to sort out the individuals on the spectrum into more definable subgroups of individuals so that we can do some of our biological tests. So basically, we took 123 ADIR items or questions, each with a severity score ranging from 0 to 3, 0 being normal, 3 being most severe, for each individual. Initially, we started with a group of almost 2,000 individuals, all with these score sheets. And then we applied several different types of cluster analyses, which are just different mathematical programs that allow you to kind of put people into different bins based upon their severity scores across these 123 items. So what you're looking at here is the results of one of the types of cluster analyses we did, where we said, after determining that an optimal number of subgroups within this 2,000 pot of individuals, there were about 4 optimally. So we asked the computer to sort these people out according to the scores on the ADIR test into 4 subgroups. And I think you can see, I hope you can see by eye, that these 4 blocks that look like plaid patterns are different. You can see that visually. And I don't have a pointer, but in that first block to the left, what you see is there's a broad red band. Let me move around a little bit. This band here. So in this block, each line represents one individual. Okay, and each column represents one of the items on the ADIR score sheet. It turns out that all of these columns refer to spoken language skills. And because they're red, and red it has a severity score of 3, which is most severe, I call that the severely language impaired group. Okay, so this is a group of severe language impaired. Here you can see there's a lot of red, comparatively speaking, in these last few columns that apply to savant skills. And because of that, there's a higher frequency of savant skills in this group. I just call them the savants. This group here, I don't know if you can tell, I hope you can, that there is more black relative to the other groups here across the board, across these items. And black equals a score of 0, which is normal. So even though these individuals are diagnosed with autism, they have a milder phenotype, so I called it mild, and the upper block there on the right, I called intermediate. But what I also did was assign color to these 4 blocks, so that when I did what's called an unsupervised clustering test, where I don't tell the computer how to segregate these individuals, I get the following result. Where down there, you can see that now each individual is represented by a single dot whose position in space is determined by the overall pattern of their severity scores across these 123 items. You can see that the severe language group, which are the red ones, they kind of segregate out towards the left. And in fact, if I rotate this, I can't do it here, but if I rotate this figure, you can see they're well separated. Okay, and then the other 3 groups here are also distinguishable. So bottom line, if you take the analogy of the fruit bowl describing individuals with autism, here we're beginning to tease apart, separate the cherries from the grapes, from the bananas, and the blueberries. Okay, so this gives us more distinct clinical subgroups to do our gene expression analyses. So we're moving up a level, 1 level beyond the phenotype. And here we're asking, what are the gene activity levels in cells from each of these individuals that have been segregated into these different clinical subgroups? And how do they compare with non-autistic controls? So I took 3 of the 4 subgroups because this was a study that was a proof of concept study, limited by time as well as money. So we have the language subgroup there, the mild and savants, and under the turquoise bar we have a series of controls, a set of controls. So there are 26 to 31 individuals per each subgroup. And what you're looking at here is called a gene expression heat map. So now we're looking at gene activity levels. And the level of the gene activity is color-coded, either green or red, according to the level of activity. So where you see green, now let me explain. Each horizontal row here represents a gene, a specific gene. Each column is an individual. So where you see green, you see that the activity of that gene is low for that set of, for this set of genes up there. It's high for this set of genes here. Well, what is clear is that there's the greatest difference in terms of the expression pattern between the controls and the severely language impaired, which is overall the most severe group among the groups we studied in autism. Then you also notice that as you go towards the milder end of the spectrum, mild and savant, you begin to see more of the color representing normal come back. So we see both a qualitative difference between autism and controls, but also quantitative difference if you look between among the phenotypes. Okay, so this is starting to get at the biology of autism. And if we do cluster analyses that's also unsupervised, based upon the set of genes, you can also segregate those individuals that are color-coded according to the gene expression profile. Here we're not talking phenotypes, we're now talking biology. So what is this biology? Okay, so now what we want to do is compare individuals in each of the clinical subgroups of autism against controls to identify which of the genes in each of the subgroups are differentially expressed. That means which ones are turned on or turned off relative to the non-autistic individuals. And this Venn diagram summarizes what we found. For the most severe language impaired group, we see over 4,000 genes that are different in activity from the non-autistic controls. For the mild group, we see 500 genes. For the savants, we have 127 genes. So what are they? What do they do? One of the things we can do here is to question what are the genes that are shared between subgroups of autism. For example, if we look at the group of 199 genes that intersect, overlap between the language and the mild, we see that the functions of those genes include genes that control cell death, inflammation, which you probably have heard in other talks, is significant in autism, embryonic development, synaptic transmission, which means how neuronal cells talk to each other, neuronal migration, how do they move about in the brain during the process of brain development, for example, genes that are involved or associated with memory and learning, muscle tone, and importantly also head size, because that was one of the very consistent anatomical observations, particularly with babies that later on are found to have autism. Also interesting are the genes that are unique to a particular subtype. If we look, for example, at the 3,900 genes that are unique to the severe language impaired subtype, we see a whopping number of cell death genes, much more than the shared ones. We see genes that are associated with painful response to normal sensory stimuli. So what does that mean? I think you all, as parents and also as care providers, you've all seen this, where kids cannot stand certain visual stimuli, certain auditory stimuli, certain tastes on their tongue or textures on their tongue, certain articles of clothing. Those are the genes. Also genes involved in muscle rigidity. I remember my son as a baby, before he had any kind of diagnosis, I recall it was really hard to kind of hold him like this because he'd always be, arching backwards and stiffening up. Then also genes that are responsible for heightened or hyposensitivity to normal painful stimuli. So either they don't react to something that's truly painful, like burning their hands, or they may overreact to something that's just very, 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 very mild. There are also genes associated with the language subgroup for epilepsy. And it turns out that language impairment and epilepsy seems to go hand in hand in autism, and I found that out later. So we're doing all of this studies with me somewhat being naive with respect to the breadth of symptoms that are experienced by individuals with autism, because my son in particular doesn't have most of those symptoms, and he's pretty healthy as far as I can tell. So I was really surprised when I found out in talking to groups like you from other parents that, they'll come up and say, well, my daughter's a cherry because she doesn't have much language, but she also has epilepsy, digestive problems, etc. And that sort of kind of wraps into that group. And then also interesting to us at the time when we first discovered this were these 20 genes at the center of the Venn diagram, because we thought that we really hit the jackpot in terms of getting an understanding of the core biology of autism. So when we looked at the identity of these genes, we were really disappointed to find that these genes are all so-called unknown in terms of coding for a protein. So these mRNA transcripts, they're produced by the cell, but they do not result in the protein, and so they're known as non-coding genes. There's now a lot more attention being paid to non-coding genes as mediators of gene regulation. So back when we published this in 2009, this was really, really novel. The interesting thing about these genes is that when we tested the activity levels of his genes, we tested 8 of them. All 8 of them responded to male hormones. So again, because autism is so male biased, we thought this probably has a biological role, but we don't know what it is yet. Okay, so where do we go next? I highlighted the term circadian rhythm. We found 15 circadian rhythm genes only in that language phenotype. So these were not altered in the other 2 phenotypes that we studied. And at the time, I was really puzzled because I thought, what is circadian rhythm, which is controlling the sleep-wake cycle? What has that got to do with autism? And then I heard a talk by a French scientist where he linked circadian rhythm to synaptic regulation, which means the control of connections in the brain, between neurons in the brain. So we started to take a closer look at the circadian rhythm genes, and I don't want to get you kind of off target with this, but just to give you an idea of how we do our studies. We took this 15 circadian rhythm genes, we put them into a program that looks at gene networks, and what it does is it searches the literature and it pulls together all the information that connects these genes to each other and to other genes and to the biological processes that they regulate. And I will come back to the gene that's encircled in red, RORA, later, and then the other thing I want to point out from this spiderwebby slide is that the red arrow pointing to steroid biosynthesis is a function we continually see no matter what kind of genomic studies we do. Okay, so here I'm summarizing that other slide. Bottom line is, what are the functions associated with the circadian rhythm genes? These include, not surprisingly, sleep-wake cycle, memory, learning, cell proliferation, and as I said earlier, steroid biosynthesis. But these genes are also associated with digestive disorders, inflammation, muscle dysfunction, and neuron toxicity. So what can you get out of this? Well, one can mine this type of information for novel target genes for subtype-specific therapies. For example, one of the genes that we saw to be decreased among the circadian rhythm genes is this gene called AANAT, which is an enzyme that's critical for the synthesis of melatonin. So this enzyme is decreased in individuals with autism within this subgroup, and its deficiency will lead to reduced melatonin biosynthesis, where melatonin is the sleep-inducing hormone. So such a finding might suggest that those individuals might be responsive to melatonin supplements. Okay, another gene that was decreased in this subgroup is this other gene called DPYD, and this gene is involved in the breakdown of DNA. It's a natural process, but the deficiency in this particular enzyme leads to reduced beta-alanine, which is one of the metabolites resulting from the breakdown of DNA. And it's also known that genetic deficiency in this particular gene predisposes to epilepsy, mental retardation, about 50% liability, and 18% of individuals with a genetic deficiency also have autism. So this apparently is a critical gene, and this might suggest that individuals deficient in it may respond to beta-alanine supplements, which is a supplement, nutritional supplement, or might also be responsive to anticonvulsant medications. Now, in today's psychiatric practice, anticonvulsant medications are frequently prescribed to individuals with autism, including my son, who doesn't have any epilepsy. But if we could identify those individuals that are deficient in this particular enzyme, then that might suggest that anticonvulsant medications might be a first-line therapy for those individuals. So all of this kind of leads us down the path of personalized medicine. which is prescribing medications based upon the specific deficits, biological deficits in each particular individual. And I mentioned also our interest in diagnosis. So without going into a whole lot of details, we wondered whether or not gene expression profiles could differentiate, distinguish individuals with autism from controls. And the bottom line answer is yes. If you look at the very last line in this table here, if we lump all of the individuals with autism together as a case group and compare them against controls, and then put our gene expression data through some programs to identify the most robust genes that can differentiate the groups, we have to use 74 genes to get about an 81% accuracy of determining which individuals are autistic and which ones are controls. If we divide them into these phenotypic subgroups, we get over 93% accuracy with much fewer genes, and more importantly, both the sensitivities of detection as well as the specificities are greater than 90%. Okay, so this is in comparison to when you lump them all together, you get a decent sensitivity but very poor specificity. So there's a company in Boston that's now working on developing this type of biomarker test for diagnosis. Okay, so far we've only been talking about that second level, gene expression profile. We wondered then whether or not the idea of subtyping might also improve the ability to detect genetic differences in individuals. So what we did was we took already published genetic data from over 4,000 individuals, cases, and 7,000 controls. We had to select out those for which there was this ADIR score sheet so we could do our subtyping, and ended up with 1,800 cases and I think 2,400, something like that, controls. But the bottom line is when you separate individuals into these different subtypes, we identified 18 novel subtype-dependent genetic variants that were not identified when people were studying 4,000 cases and 7,000 controls. So this shows the increased power that is derived from segregation, according to clinical phenotypes, in the ability to detect genetic variants for autism. Now, what are some of these variants? It turns out that all of these 18 SNPs, so single nucleotide polymorphisms, these are single base changes, are all in the non-coding regions of the DNA. Okay, so they're not likely to change the structure of a protein. More likely, they might change the regulation of the protein, whether it's turned on or turned off, or how it's kind of spliced together. All right, but some of the genes that are associated with these non-coding regions are ones that make sense in the context of what we know of autism. What's missing up there in the black curtain are several genes, which are the serotonin receptor 4 and 2 chemokines that are implicated in gut disorders. Okay, so that tells us something about the biology, too. And then that other gene, GCH1, is an enzyme involved in tetrahydrobiopterin folate metabolism. And we know that the folate pathway is important in development. If we do a functional analysis of the genes, we see that these genes converge on pathways that lead to axonogenesis, which is kind of the extension of neurons in order to migrate and touch other cells, neurogenesis, the formation of new neurons, again, steroid metabolism, long-term potentiation, which is involved also in memory and learning, as well as cell adhesion, which is important for neuronal migration, moving about in the brain, in brain development. Okay, so we've covered now genetics. Let's move on to the other side, epigenetics. Question, why study epigenetics? Well, epigenetic changes, aside from genetic changes, epigenetic changes may also be heritable. Okay, so some people say that epigenetics may be the one source of missing heredity in autism, and it has not been well studied. The other point about epigenetics that I made earlier is that epigenetic changes are reversible because you're just putting a tag onto the DNA which can be removed. Methylation is such a tag. But then there are other mechanisms of epigenetic regulation that also can be altered, all right? When you have a change in the DNA, that is harder to fix. The third point about epigenetics is that environmental agents often work through epigenetic modifications. So there could be, if we're looking at the top of the pyramid, there could be agents in our environment that could alter our genome in a heritable manner. That means it can be passed through the generations by inducing epigenetic changes. Okay, so I point this out because we studied two of the mechanisms for epigenetic changes in autism, including DNA methylation and microRNA expression. So the way we did these studies is we had a set of monozygotic identical twins and also SIB pairs, because this helps to level the genetic playing field. Identical twins have identical DNA sequence, so we don't have to worry about differences there. And siblings are also should be pretty similar, but we know they're different, but they're also similar. So that helps to level the genetic playing field. Then we did global methylation analysis on a genome-wide scale. That means to look at all of the different genes that are differentially methylated, that have this chemical tag put on in autism, in the autistic individual versus the control. And we overlapped that with the data we got from gene expression studies for the same individuals. And what popped out were a set, for methylation studies, were a set of about 20-something different genes where there was increased methylation and reduced expression, which is the relationship you would expect between methylation and expression. So higher methylation, reduced expression. One of these genes is a gene never before associated with autism called RORA, for retinoic acid-related orphan receptor alpha. That's a mouthful, so we just call it, it's just RORA. Now, why is RORA interesting? And why might its deficiency be relevant to autism? We know most about RORA from what is known about it in the mouse models for RORA deficiency. We know that this gene, RORA, and it's lowercase when you're talking about an animal, uppercase for humans, that we know that RORA protects the brain against oxidative stress and inflammation. It regulates circadian rhythm, or the sleep-wake cycle. The loss of RORA leads to developmental defects in the cerebellum, which is like in the back of your head here, as well as the loss of Purkinje cells, which are the very large cells in the cerebellum. Furthermore, RORA-deficient mice exhibit sex differences with respect to production of cerebellar neurosteroids and Purkinje cell survival. So the bottom line is there are sex differences. We know that autism, is manifested by an overwhelming, a disproportionate amount of boys affected than girls. Okay, finally, although most studies on the RORA-deficient mouse involves studies on ataxia, which is lack of motor coordination, and hypotonia, muscle weakness, there's been reports that the behaviors of this mouse model, to me, are reminiscent of autism, including them exhibiting preservative behavior as well as deficits in spatial and discrimination learning. In other words, they can learn okay, but when you change the conditions, it's hard for them to unlearn what they learned and to adapt to the new set of conditions. So it's sort of like adaptive behaviors. Now, how does it relate to autism? Well, studies on autism, on the autistic brain, have shown evidence for neural inflammation as well as oxidative stress. Circadian rhythm genes are implicated not only by our studies, but also some genetic analyses where some of the circadian regulators have been found to have genetic variants. It's been well known for many years, and this is the most consistent neuroanatomical abnormality. It's been known that Purkinje cells are deficient in the brains of individuals with autism. And this RORA is very important for the survival of Purkinje cells. And then finally, males are more affected in ASD than females. So what are the questions regarding RORA's involvement in autism? So bottom line is we want to know what are the regulators of this gene called RORA, what are its targets? And I say targets because RORA is one of those genes that itself regulates a whole bunch of other genes. It's a nuclear hormone receptor that has the property of being able to turn on or turn off other genes. So what are they? So here are some of the specific questions we're interested in. Is RORA differentially regulated by male and female sex hormones? Is the deficiency evident also in the brain tissues of individuals with autism? Up until now, I've been talking about the data that we've learned from studying the blood cells of individuals with autism. Finally, what genes are in turn regulated by RORA, and how do they relate to autism? Okay, first of all, the hormones. We found that in a cell line, the RORA is oppositely regulated by male and female hormones, with female hormones, the upper two blocks, showing an increase in the level of this gene RORA, and excuse me, in the lower two blocks, testosterone, or a form of testosterone, decreases the level of RORA expression. So we see this opposite effect of male and female hormones. What does that mean? Okay, then we're also wondering, is RORA reduced in the autistic brain, and which autism relevant genes are regulated by RORA? One of the clues to this last question comes from the cancer literature that reported that RORA regulates a gene called aromatase in breast cancer cells. So in this slide here, we're looking at the levels of RORA protein and aromatase protein in the brain, postmortem brain tissues of individuals with autism versus non-autistic individuals. And here you can see in the left block that there is a significant decrease in the level of RORA protein as well as the level of aromatase protein. The other two bars, DAPI, is just a control for nuclear staining, and there's no significance there. And if you compare the level of aromatase protein with that of RORA protein, you have almost a one-to-one correlation. That would be supportive of the idea that aromatase is one of the target genes regulated by RORA. Okay, by now I think you're thinking, so what? All right. So what is the relevance of the decrease in aromatase? Well, getting back to the male-female ratio, the autistic male-to-female ratio is at least 4-to-1, and it may be higher when you get towards like the Asperger's end of things. Simon Baron Cohen from Cambridge has proposed the extreme male brain theory, which is summarized below as elevated fetal testosterone is a risk factor for autism. However, it's a big black box connecting the two. Okay, so there's no molecular explanation for how higher testosterone levels might be a risk factor for autism, and whether or not this accounts for the significant male bias. Well, what is the function of aromatase? Aromatase is an enzyme that converts testosterone to estrogen. All right, so therefore, in autism, where you have a decrease in RORA and a decrease in aromatase, this would lead, if you block that enzyme, you could see that it would build up in testosterone. You cannot convert testosterone to estradiol. So you'll have an increase in testosterone, decrease in estradiol, which led us to propose this mechanism for testing. And that is, we've shown that DNA methylation can reduce the level of RORA, and the reduction in RORA reduces aromatase, which leads to an increase in testosterone. And we've shown that testosterone further can decrease the level of RORA. So this is sort of a cyclic effect, that you have decreased RORA leading to increased testosterone with further increases RORA. So it's an exacerbating effect. On the other hand, for females who have a higher amount of estrogen, we've shown tends to increase the level of RORA expression, thus protecting females a little bit from conditions that would lead to RORA deficiency. So the last question I'm going to address here is RORA a master regulator of other autism candidate genes? So we looked at this question on a genome-wide level. So we looked at we considered all of the possible genes that are synthesized in our bodies, and which of these could possibly be regulated by RORA. And I won't get into the technical details, but what we found was that there are over 2,500 potential target genes of RORA. And of these, over 400 are already known as autism candidate genes. So they're in the autism gene databases. These genes are highly enriched for neurological functions, including neurogenesis, synaptic transmission, plasticity, axonogenesis, cognition, learning, and memory. Then, it's not sufficient to use one technique and identify. You have to validate it. So we followed up on these studies by doing functional knockdown. So we knocked down the level of RORA and looked at the level of expression of these 6 targets of RORA, validated all of that. And we also showed that these 6 genes are reduced in brain tissue samples that are deficient in RORA, further supporting the fact that these genes are transcriptional targets of RORA. In other words, they're regulated by RORA. So what are the functions of these genes? Well, they're involved. This one here is involved in synaptic transmission, excitation, and importantly, developmental delay. This gene, aromatase, we've met before, involved in neurogenesis, plasticity, but also protects against oxidative stress, is involved in social cognition. This gene, again, also converts testosterone to estradiol. So both of these enzymes that do that are regulated by RORA. It's involved in mitochondrial integrity, but also to X-linked mental retardation associated with language impairment. This gene is involved in synaptogenesis, plasticity, dendritic contact, in other words, neurons reaching out, touching each other, long-term depression. Again, another gene involved in cellular adhesion and neuronal migration, but also repetitive behaviors, spatial memory. So we're getting to the high-level functions associated with these genes. And finally, this other gene is involved in axon guidance, leading neurons to where they're supposed to be, synaptic plasticity, mood disorder, and learning. So the bottom line here is we've already validated at least 6 of these target genes of RORA, all of which have been shown to be involved in the pathological processes of autism. So the take-home message here, I think, is that I think that any mechanism that disrupts RORA expression, including genetics, epigenetics, or environmental factors, may increase the risk for autism. Okay, so is it a target? Is RORA a target for gene-environment interactions? It's the first candidate gene for autism demonstrated to be oppositely regulated by male and female hormones. And the impact of sex or endocrine hormones on this critical candidate gene for autism suggests that a wide range of environmental compounds known as endocrine disrupting compounds may lead to increased risk for autism through gene-environment interactions through RORA. So I'm happy to say, well, first of all, what are examples of EDC? Some of them are very familiar to you. One of them is atrazine, which we're studying in my lab right now. It's found in virtually all herbicides. Bisphenol A, or BPA, you're all familiar with, is coming out of plastics or paper receipts. Phthalates in soft toys, fluorine material, polychlorinated biphenyls, valproic acids. So there are certain medications that fall into this category of being endocrine disrupting chemicals as well. And so I'm happy to say that my lab has just received funding from NIEHS, Environmental Health Sciences, to study the connection between this type of chemicals and the gene RORA as a possible risk factor for the increase in autism that we're seeing today. Okay, so then to quickly summarize, what we've done is to break down autism into these different subtypes and then associate different phenotypes of autism to distinct gene expression profiles, which gives us a little bit of an idea of what the biology is behind each of the subtypes. And then we integrate it too. We integrate gene expression profiling with epigenetics to learn a little bit more about gene regulation. We study genetics also by phenotypes and have shown that by doing so you get a lot more out of the genetic data than if you don't. And then finally we're looking now at various environmental triggers that could increase the risk for autism. Okay, so this is the take-home messages for all of my talk. That is that gene expression profiling that associates biological deficits with ASD subtypes shows the potential for targeted subtype-specific therapies. I've shown you that we can get fairly high accuracy of separating autism from from non-autistic individuals using gene expression profile, which shows the potential for developing a biomarker screen for autism. The involvement of epigenetic changes opens a new avenue for treatment based upon epigenetic regulation or reversal. Molecular alterations in blood-derived cells are reflected in brain tissues of autistic individuals, linking blood to brain. Now, this is important for two reasons. First of all, I think it shows, the fact that we see similar changes in the blood shows that there is likely systemic deregulation of certain critical functions. And secondly, it also validates the use of peripheral tissues like blood cells, blood lymphocytes, as an experimental model for autism, because God knows it is so hard to get brain tissues. And finally, it goes without saying that understanding the biology of autism will lead to more targeted and personalized therapies to correct deficits. Okay, and then I can't conclude without thanking all of the people in my lab. They're not there now, but they've contributed greatly to different components of what I've talked about today. And then last but not least, I want to thank my son Matthew for inspiring me to enter this research field to begin with, which for me has been the most exciting thing I've ever done. Thank you very much.
Thank you, Dr. Hu.